Ever wondered how some plants just seem to thrive, even in really tough conditions where others just can't? What if a big part of their secret is hidden deep in their DNA? Like, specifically, how many copies of their genetic instruction book they actually have? Today, we're diving deep into polyploidy. It's this fascinating biological thing where organisms have more than the usual two complete sets of chromosomes, and it's fundamentally shaped how plants evolved and adapted over time.
Yeah, our mission today is really to unpack this whole world of polyploidy. We'll explore what it is, look at the surprising advantages, but also the challenges it brings, and how it affects everything from those giant desert cacti to, the crops we eat every day. We'll even touch on how it might help plants deal with, well, climate change.
And we've dug through a whole stack of sources for you, everything from detailed research papers on specific plants to broader genetic overviews. We're pulling out the key stuff, most important bits of knowledge. Okay, so let's unpack this term, polyploidy. At its heart, it just means having extra sets of chromosomes, right? So instead of the usual two sets diploid, you might get three, four, six, maybe even more.
Yeah, exactly. And, how it happens is pretty interesting. It's often down to what scientists call a mitotic or meiotic catastrophe.
Hmm.
Basically, an error during cell division, something like non-disjunction, where chromosomes just don't separate properly. This can lead to making gametes, the reproductive cells, sperm or egg, that accidentally have a whole duplicate set of chromosomes. So for example, if one of these double-set diploid gametes fuses with a normal single-set haploid gamete, you end up with a triploid. Those are often unstable, maybe sterile. But if two of those diploid gametes manage to fuse, then you can get a stable tetraploid with four sets.
Right, so these errors can lead to different outcomes. And I know we often talk about two main types of polyploids. Could you break those down quickly?
Sure. Yeah, we generally distinguish between autopolyploids. That's when the genome of just one species gets multiplied, like it just duplicates itself within that single species.
Yeah.
And then you have allopolyploids. These are a bit more complex. They happen when two different species hybridize, and then that combined set of genomes gets doubled.
It's really striking how common this is in the plant world compared to, say, us animals.
That really is a key point. It's super rare in most animals. I mean, polyploid human embryos, sadly, almost always result in spontaneous abortions. It accounts for maybe 10% or so of early miscarriages. But in plants, incredibly common. Over half of all flowering plants are clearly polyploids, and many others show signs of ancient polyploidy events deep in their evolutionary history. It just tells you plants are remarkably tolerant of these massive genetic changes.
Okay, so here's where it gets really fascinating. Having all this extra genetic material isn't just some weird anomaly. It often gives plants some pretty amazing advantages that can drive evolution forward.
Absolutely. One of the biggest ones is something called hybrid vigor or heterosis. Polyploid offspring can sometimes just be way more vigorous, healthier, bigger than their diploid parents.
Mm-hmm.
And part of the reason connecting it to the bigger picture seems to be this buffering effect. Having extra gene copies kind of shields the plant from the harmful effects if one copy has a bad recessive mutation. And in those allopolyploids, the ones from hybridization, it can help maintain lots of genetic diversity, heterozygosity across generations. That's a powerful evolutionary tool.
So it's like having a built-in backup system, or maybe even multiple backups for really critical genes.
Precisely. And that gene redundancy, having spares, also opens the door for diversification of gene function over time. Think about it.
Yeah.
An extra gene copy that isn't absolutely essential for the original job is sort of free to mutate, potentially evolve a completely new function. That creates new possibilities for natural selection to act on. Polyploidy can also shake things up reproductively. It might disrupt self-incompatibility systems, meaning a plant can suddenly self-fertilize. Or it might favor asexual reproduction, which could be great in stable environments. We often see it linked with adapting to big environmental shifts, even things like plants adapting to urban environments.
But okay, like any superpower, there's usually a catch, right? There must be downsides or challenges to carrying all this extra genetic weight.
Well, definitely. There are several hurdles. One really fundamental issue is about basic cell geometry, believe it or not. It's the balance between the cell's volume and the surface area of its nucleus. When the genome doubles inside the nucleus, the volume the chromosomes take up roughly doubles. But the surface area of the nuclear envelope, the skin around the nucleus, only increases by about 1.6 times, so things get cramped. This can disrupt the delicate exchange of molecules between the chromosomes and the rest of the cell. And then there's the mechanics of cell division itself. Polyploidy can complicate both mitosis and meiosis. you see more errors, like spindle irregularities, which can lead to aneuploidy cells ending up with the wrong number of chromosomes, which is usually bad news. This is especially tricky during meiosis, making reproductive cells, when you have more than two sets of chromosomes trying to pair up and separate correctly. For polyploids in particular, meiosis can get really messy, leading to abnormal segregation and mostly aneuploid gametes. And finally, you can get gene expression changes that aren't simply proportional to the extra gene copies. Things get out of whack. Plus, epigenetic instability. Polyploidization itself can trigger widespread changes in how genes are regulated, even without changing the DNA sequence itself. These are particularly strong in allopolyploids, where merging different genomes creates what's called a genomic shock.
Okay, let's try and grind some of these complex ideas. Let's talk about a specific, really cool example. The giant cardon cactus, Pachycereus bringlei. It's that massive cactus you see towering over the Sonoran Desert in Mexico and Baja California. And it's known for having really diverse ways of reproducing.
Right, and there was this recent deep dive study looking specifically at the cardon. They wanted to see if its huge geographic range and its different reproductive strategies were linked to its ploidy level, its chromosome count. And they found something pretty remarkable. For the very first time, they actually found diploid populations of this cactus, normal two sets of chromosomes, 2N and 22. But here's the kicker. They were found only on two remote deep water islands in the Gulf of California, Catalana, and Cerralvo Islands. And these island populations, they had the ancestral hermaphroditic breeding system, meaning flowers with both male and female parts. Now, contrast that with the cacti found all over the Baja California Peninsula and mainland Sonora. Those were all tetraploid, 4 sets of chromosomes, 2N44. And these widespread mainland populations showed non-hermaphroditic systems. Some plants female, some male, sometimes a mix.
Wow. So this cytogeographic distribution, the link between chromosome number and where they live, what does that tell us about the cactus's history?
Well, if you connect the dots, it really strongly suggests that those diploid hermaphroditic island populations are like living relics. They represent the older ancestral state of the species. The polyploid condition, the tetraploid with separate sexes, seems to be derived, it evolved later. And it looks like that jump in chromosome number, the polyploidy event, was probably a key factor. It seems to have enabled the cardon cactus to spread out, colonize much wider areas, move northward. It shows a really clear link between ploidy, how it reproduces, and where it can live.
That makes sense. Were there other differences they could see between the diploid and polyploid cacti, besides the chromosomes and where they grew?
Yes, that's another interesting bit. Even though, just looking at the main cactus body, they look pretty similar. Researchers found subtle but statistically significant differences in their flowers. For instance, the diploid flowers from those islands had wider nectaries, the parts that hold the nectar, and they produced larger amounts of pollen compared to the tetraploid ones on the mainland. Now, these might sound like tiny details, but flower characteristics are crucial for attracting pollinators. So these subtle differences could actually help keep the diploid and polyploid populations reproductively isolated from each other.
So what does this all mean for how scientists might classify these cacti now? Does this change things?
Yeah, it definitely raises an important question for the taxonomists, the people who classify organisms. Given you have these consistent differences in ploidy level, where they live, how they reproduce, and even these subtle flower details, the researchers are actually proposing that these diploid island populations should maybe be recognized as a new taxonomic entity. Specifically, maybe a new subspecies, not just a different cytotype or chromosome variant of the same species. It builds a really compelling case that polyploidy, along with these other factors, directly drove the formation of a new lineage, essentially speciation, in this iconic cactus.
That cardon story really drives home the direct impact polyploidy can have. But its influence goes way beyond desert plants, doesn't it? It reaches right into our farms, our food, and maybe even the future of plants on a changing planet.
Absolutely. I mean, polyploidization has been a workhorse in plant breeding and crop improvement for ages. Breeders have used it, sometimes intentionally inducing it, to boost yield and biomass in crops like potatoes, red clover, sugar beets, even watermelons. And recent studies, like some really detailed work on Arabidopsis thaliana, that's the little lab rat of the plant world, give us great insights into how this works. Researchers created a whole series of Arabidopsis plants with different ploidy levels: diploid 2N, tetraploid 4N, hexaploid 6N, and octoploid 8N. And they found polyploidy had huge effects on growth and, interestingly, cell wall makeup. for example, the Tetraploid Arabidopsis plants, they developed way larger rosettes, the leafy base, like a stunning 250% bigger area than the normal diploids, and their stem dry weight shot up by 82%. Significant increases.
Wow, 250% larger.
Yeah.
That's huge. But did that trend just keep going up? Like, were the octoploids even bigger?
Interestingly, no, it kind of peaked. The hexaploids showed some increase over diploids. Yeah, but the octoploids didn't really gain much or even decrease in some aspects. It illustrates something people sometimes call the high-ploidy syndrome. Austin, it seems beyond a certain point, the benefits of having larger cells get outweighed by a big slowdown in how often the cells divide. So overall biomass production can actually level off or drop. But here's something really exciting, especially thinking about the bio-based economy, biofuels, biomaterials. It's polyploidy's effect on cell walls. That Arabidopsis study found something fascinating. As the ploidy level went up, there was a gradual decrease in things like lignin and cellulose. Those are the really tough, rigid parts of the cell wall that are hard to break down. But at the same time, there was a positive correlation with matrix polysaccharides, stuff like hemicellulose and pectin, which are generally easier to process. This shift in composition dramatically improves saccharification yield, which basically means they could get more fermentable sugars out of the plant biomass much more easily, especially if they use some chemical pretreatment. So inducing polyploidy could actually be a strategy to engineer crops, specifically for better biofuel or biomaterial production. Pretty cool, huh? And connecting back to adaptation, polyploids generally seem to show increased phenotypic plasticity. That's the ability of one genotype to produce different traits depending on the environment. It lets them be more flexible, more adaptable. So you often find polyploid species doing well in tougher spots, like drier regions or higher altitudes. Now, it's not a universal law. Other things like the specific genetic background matter a lot. But polyploidy is definitely a major tool in the plant adaptation toolkit.
So taking all this together, what's the big picture takeaway for how we understand plant evolution? Are polyploids just always the evolutionary winners? Is it that simple?
Yeah, that's an important question to ask. And the answer is, well, it's nuanced. While polyploidy is undeniably a major driver of diversification in most green plant groups, there aren't many hard and fast universal rules. For instance, look at gymnosperms, conifers, cycads, that group. Polyploidy is notably rare there, yet they're a hugely successful and dominant plant group. So success doesn't always depend on doubling genomes. Even our major food crops show a mix. Wheat is hexaploid, rice is technically diploid but shows signs of an ancient large duplication. Maize is an ancient tetraploid, sugarcane is a complex high polyploid. It just highlights that evolution uses diverse strategies. There are many paths to success. But you know, what really stands out to me, especially when you think about the challenges ahead climate change, increasing human impact, there's this potential for what some have called an explosion of polyploids. If you look back at Earth's history, periods following mass extinction events often saw the appearance of many new polyploid species. That ability of polyploids to rapidly generate new gene combinations, to adapt quickly, it might become an even more critical strategy for plant survival and diversification in the future we're facing.
Wow. What a truly fascinating deep dive into this complex, sometimes counterintuitive world of polyploidy. From controlling how big an Arabidopsis rosette gets to allowing a desert cactus to conquer new territories and even split into new lineages, it's clear that the number of chromosome sets inside a plant cell has profound consequences for its entire life.
It really is a testament to the incredible flexibility, the adaptability that's encoded right there in life's book of instructions. Polyploidy is just such a great reminder that evolution is always tinkering, always experimenting, finding new ways for species to thrive, even when facing really significant challenges.
So what does this all mean for you, listening in? Well, next time you look at any plant, could be a giant tree, a tiny wildflower, even the vegetables on your dinner plate, just take a second. Consider the hidden genetic story inside its cells. How many chromosome sets might it be carrying? And what incredible story of adaptation, resilience, and evolutionary journey might those numbers tell? Thank you for joining us for this deep dive.